So thank you for joining us today for our virtual lecture series, which is part of the Graduate Admissions Department here at Halmos College of Arts and Sciences. My name is Farah Amro. I'm one of the graduate admissions counselors at Halmos College, and joining us as well from the Department of Biological Sciences is Dr. Christopher Blanar, our presenter for today's lecture. And from the graduate admissions team as well, Mrs. Megan Choi is joining us. She's the Assistant Director of Graduate Admissions at our college. Mrs. Megan Choi is the assigned graduate admissions counselor for the Department of Biological Sciences. So actually, she's your best person to contact for any admission inquiries. However, all of us here in the graduate admissions department are happy to help you if for some reason you're unable to reach Mrs. Choi. Dr. Blanar's lecture today will run for about 45 minutes, which after we will have about 15 minutes left for our audience to ask any questions they might have regarding today's discussed topic, admissions requirements, or NSU in general. And to control the flow of the lecture and respect your precious time and Dr. Blanar's time, we ask you to please write your questions as they come to your mind in the chat box. During the Q&A session, I will read your questions to Dr. Blanar or to Mrs. Choi to answer them for you. A little bit of a background about our awesome presenter for today, Dr. Christopher Blanar. He's an associate professor at the Department of Biological Sciences at the Halmos College of Arts and Sciences. Dr. Blanar moved to Florida from Montreal in 2011 after stints at Environment Canada and Bishop's University in Quebec, as well as SUNY Oswego in New York. He held adjunct positions at McGill University, Concordia University, and the University of New Brunswick at St. John. At NSU, Dr. Blanar directs undergraduate and graduate research projects. His research combines parasitology and general ecology. He does parasite surveys of wildlife, especially of fish and birds, and studies the processes that structure parasite communities. And as part of his research activities, Dr. Blanar also tracks changes in parasite communities as markers of ecological health. His projects typically involve some combination of field work in critical Florida habitats, such as the Everglades, mangroves, swamps, coral reefs, and sargassum mats, as well as laboratory-based work, which takes place on NSU's main campus in Davie, Florida, or in partnering labs in our state-of-the-art oceanographic campus, which I'm located in right now in Dania Beach, Florida. In addition to all this, and through research partnerships with faculty members at NSU and beyond, Dr. Blanar also conducts research on the biocontrol of parasites, parasite-induced host behavior modification, and parasite phylogeny. All this work leads to today's lecture, during which Dr. Blanar will talk to us about adventures in parasitology, including hosts, parasites, and other unexpected relationships. Dr. Blanar, thank you so much for taking the time today to discuss some of your life's research work with us. You have the stage now to start your presentation. Thank you so much.
And thank you so much for the invitation. I'm going to be sharing my screen now. I have to do the obligatory thing where I ask everyone if they can see it.
Yes.
All right, perfect. Thank you. All right, so the title of my talk today is Host Parasites and Other Unexpected Relationships. And my goal with this talk is to give you an overview of a little bit about my pathway and how I got to where I am now, and talk a little bit about the work that I'm doing now and some of the interesting opportunities that work has generated for some of the students that have come through my lab and partnering labs. And so without further ado, I'm going to launch into this talk, which is titled Adventures in Parasitism. Ms. Farah Amro, it just gave such a great introduction to my life that I scarcely feel the need to repeat it. The only thing I'll point out here is that everything about my CV points to an orderly progression of starting off doing a Bachelor of Science in Biology, Ecology. I'm split between McGill University and Concordia University, through a Master's in Natural Resource Sciences at McGill, to a PhD at the University of New Brunswick and St. John, to a fantastic 3-year NSERC postdoctoral fellowship. And it all looks so linear, and I really need to tell you right now, as prospective students, that nothing could be further from the truth. that I have followed the most incredibly non-linear pathway. And don't be fooled by people's CVs, especially when you see a CV like this. There's absolutely nothing about my career path that was linear. And so that's why my first slide is a quote from the Joker, which goes, "Do I really look like a guy with a plan?" At no point have I really had what one could call a plan. I have had students come to me say, "How did you plan your career?" And I always feel bad trying to give that advice to people like that because I really didn't. Um, in fact, as a kid, I kind of dreamed of being a vet. But I ended up getting into biology at McGill. And then through a bunch of chance meetings, as you'll see, I ended up getting diverted from that path. And since then, as you'll see, I've followed consistently the path of most interesting opportunity and most fun. And that has ended up working out in fact quite well for me. So I'll start a little bit by talking about how I got my start. So I started off as a Bachelor of Science student at McGill University, and I needed to get a field course under my belt, and all of the other field courses were full, and there was this one that was taking place in this location here, which is a place called La Cartelle, which is in the eastern townships near Montreal, Quebec, Canada, where I'm from. And and another reason for me to go was that there was a girl I was crushing on that was taking that class too. So I was like, okay, multiple reasons to go. So I went and I took this class, and I registered for that class for kind of all the wrong reasons. I kind of had to, and so on. But it ended up being an absolutely game-changing experience for me because while I was on this field trip, I first interacted with a professor named Joe Rasmussen at McGill. So Joe Rasmussen is the person on the left here. Joe is, I guess, science famous for being one of the big pioneers of the use of stable isotopes in the study of ecosystems. And so Joe, and also the study of contaminants and pollutants in ecosystems as well. And so Joe had a really busy kind of interesting lab. He had a student named Gilbert Cabana, who's the dude in the middle there holding up the fish. Gilbert is now at the University of Quebec at Trois-Rivières as a faculty member. And he also had another guy, Paul White, who's the person on the right, who's a professor at the University of Ottawa. So all these people are doing interesting things now. And those three guys were at that field trip, and I had never had a chance to sit down and talk to people about science and just talk about the practice of science and talk about biology and talk about ecology and the relationships between organisms and some of the cool things and interesting things that happen when you study them. And so by virtue of being in this field trip and concentrated with them in this one spot for 2 weeks, I got incredible access to them. And those were some of the most interesting and stimulating conversations I'd ever had in my life. And I'm sure these guys don't even know how much of an influence they were. But by the end of that week, all sort of thoughts of being a vet were out the window. And I wanted to be these guys. I wanted to interact with people like these people for the rest of my life. I wanted to have those kinds of conversations for the rest of my life. It was absolutely game changing for me. And a lot of that was cemented when, through conversations with them, I took on a field project looking at parasites of these small pike. This is a fish that you find in northern lakes, not so much down here. And the pike, where the field course were absolutely riddled with these black dots, almost like someone had sprinkled peppercorns on them. And each of those little black dots is a small parasite. It's called black spot disease. The parasite that causes it is something called uvulopher ambloplitis. And um, we were looking at these fish, and I asked Joe and I asked Gilbert, does it hurt the fish? Surely it hurts the fish, this massive infection. And they were like, I don't know, nobody really knows. So I decided right then and there that was going to be my field project. And I did my field project on that. And as it turns out, the parasite doesn't harm the fish at all. We couldn't demonstrate any measurable harm to the fish at all, even at the incredible infection levels that you see on this fish. I mean, look at this thing, right? You can see the natural spotting of the fish sort of around here at the top, but all those black dots along the side, the deeper black dots are all parasites, and yet no effect, right? So after my undergrad, I was casting around for things to do, and I didn't really have much of a plan. And I was working at a place in Montreal that was kind of like a zoo. And one of my good friends at that place was doing his master's at McGill, and he said, "Hey, there's this cool guy named Mark Curtis that you should do a research project with." And I was like, "Oh, what does he work on?" He was like, "Oh, parasites," and that brought me back to that cool project that I'd randomly done in La Cartelle as an undergrad. I was like, "Okay, maybe I can do that." So I sat down with Mark. We hashed out a project. And thanks to that project, I ended up working with Arctic fish. I ended up spending time in the Canadian Arctic in a small community called Igloolik, which is indicated on the map on the left. And working at the Igloolik research station, which is that spaceship looking thing on the right. And this project involved, oh yeah, by the way, folks, I'm a parasitologist and I'm going to be talking about my research. And that means that by definition, some of the pictures I'm going to be showing you are going to be of hosts, in fact, with parasites, and there's going to be some dissected fish, and I think one dissected iguana later on. So just be aware that that's something that's going to come up in the talk, and prepare yourselves. So yeah, I traveled up to the Arctic and I sampled some fish and I sampled some shrimp. And the parasite that I was working with was a tapeworm, an Arctic tapeworm that is now called, it used to be called Diphyllobothrium dendriticum, and it's been since reclassified or reclassified as something called Dibothriocephalus. So it's the name's changed, but it's still the same parasite. So on the right here, you see an image of the adult parasite. This is something that you'd find in dogs or foxes or even in fish-eating birds. And you can see that it's a typical tapeworm. It's super long and thready and so on. And so the birds, for instance, will shed eggs from the tapeworm in its poop into the water. Those eggs will hatch and move into the little shrimp that you see on the left here. So this is the parasite inside the shrimp. And then this shrimp is going to get eaten by a fish. And you can see a fish can become heavily infected. Every single one of those like pimple-like things on the surface of the intestines here is a little parasite waiting to be eaten by a bird. And the question that the First Nations the project was partially funded by the First Nations, and the question the First Nations people in Igloolik had was, is this negatively affecting the fish? So I had the prior experience thanks to my random undergrad project. So I went in and did the project, and I had all these ideas about how the fish were going to be negatively affected by the parasite because the infections were so heavy and the viscera get all twisted and stuff like that. And yet I could not demonstrate any harm to the fish. And in fact, I also looked at other, like in combination, parasites in combinations with other stressors, such as pollutants, and I could not find any effect on the host. Right. So it's kind of interesting that even massive infections like this of a parasite, I could not find any physiological effect on the host or any effect on the nutritional quality of the meat that they were getting from these fishes and so on. So yeah, it looks gross, but from a nutritional perspective, just sort of eat around them and you're fine. And you're fine. And considering that the parasite's mostly on the guts anyways, it's probably less of a problem than it looks. I wouldn't eat this fish raw, right? That's you might get the tapeworm, and in fact, many people do. And we can talk about that later in the question period. There's usually a few questions about stuff like this. Um, and we could talk about this life cycle because it's such a cool life cycle. And people do get it. If you eat this fish raw, if you were to eat this fish raw, you'd be at risk at least. Okay, so I personally found no effect on the nutritional quality of the meat or the physiology of the fish or anything like that. All right, so that was my master's. So then I was sort of casting around, sort of thinking, well, what am I going to do? And I had another job. I worked for the private sector for a little bit, counting eggs and things like that for environmental impact assessments. And then just through a chance meeting I went through a guest lecture at the university. That someone turned me on to the idea of going to the east coast of Canada, to New Brunswick and doing my PhD there. So I ended up going to the University of New Brunswick and working with two wonderful people, Deborah McClatchy, who is now the president of Wilfrid Laurier University in Canada, and so she's moved on to amazing things. And Kelly Muntrick as well Kelly McKittrick as well has moved on, and he's now a professor and a researcher in Alberta, I think, at the University of Calgary. So both of them have moved on, but at the time they were faculty at the the University of New Brunswick at St. John. And you can sort of see New Brunswick and St. John in the background, where in the Kelly's picture. And so my PhD project was sort of like a field biologist dream in many ways. I'd gotten my start doing crazy field work with fishes, and this continued that trend, from the Lac Ertel to the Canadian Arctic to some random streams in Quebec to now some beautiful, absolutely beautiful salmon-bearing streams in throughout New Brunswick. And the the project was really cool because basically what we were doing was looking at the effect of a mine. There was a copper and zinc mine at the top of the watershed, and effluent was coming out from the stored waters adjacent to the mine and going into the streams. And the idea of the project was to look to see how that effluent was affecting the ecology of the streams downstream from that location. And my idea was, well, maybe the effluent from that mine is hitting the fish and it's irritating the gills, or it's irritating the skin of the fish, stressing them out. And that in turn is making them more likely to get parasites. And so the whole idea was increased. My hypothesis was that I was going to see increased parasitism downstream of the mine. And so it was wonderful because for 3 years I got to travel throughout the late summer and early fall in some of the most beautiful rivers in Canada, these beautiful streams like the one that you see up at the top there. And I got to do this really cool thing. If you can look at the image in the top left there, and you see I'm wearing what looks like a backpack with a car battery strapped to it. And essentially the way the crazy apparatus works is that you introduce an electrical charge into the water, and that doesn't kill the fish, but it definitely stuns them enough that you can then more easily catch them in nets, like you see my colleagues doing just upstream of me here. Um, and or actually downstream. They're actually so slightly downstream of me. And the idea here is that salmon and the salmon that we were trying to collect, these are Atlantic salmon, Salmo salar. Uh, the the salmon like to hide out under the gravel, and that makes them difficult to catch. But when you zap them, they kind of twitch their way out from under the gravel, or they float from under the gravel, and then that makes them easier to catch. And so we would catch them that way. So this technique is called electrofishing, and then we'd put the fish in a bucket to recover. I would do lengths and weights. And once the fish were more or less recovered, I would examine them visually. And essentially, it involved sort of picking up the fish. Look how beautiful these fish are. I mean, really. Um, yeah, they're absolutely stunning. I loved working with these fish. And you can see that this is from a dissected gill, but you can see these are the gill filaments here in the lower right. And you can see these large worms, this large worm sort of clamping onto the gills. And this worm that I was studying is a gill worm called Discocotyledon sagittata. And it sucks on the blood of the fish, and it's a known pathogen of the fish, especially at high densities. So it was really interesting for me to see, well, are there changes in Discocotyledon sagittata as the gills get irritated, as the fish gets irritated? Does that weaken the fish's resistance and natural resistance to these parasites? And so do we see more of these parasites in waters that are downstream of the mine? So that was the whole idea, that increased parasitism. And so this is the result from that study, and I'll just walk you through the design. So this big red blotch in the upper part where it says Little South Branch, that's where the mine is. And then all of the rivers sort of adjacent to that are affected in one way or another by effluent from that mine. So you can sort of picture anything in the red zone. My sample sites are labeled with numbers. They're D1, D2, D3, U1, and so on. And you can imagine that the effluent concentrations are really high, sort of right close to the mine. But then as you move further and further downstream towards the more distant sites like D4, D5, and DR2 at the bottom, the concentrations of metals decrease as you move downstream. And then, of course, we had to have some control rivers. So we had some adjacent rivers, the Northwest Miramichi River, absolutely gorgeous. We had a bunch of sites there. And then the North Branch and the South Branch of a river called the Big Savogle, absolutely beautiful as well. Hold on, just going to dismiss this. Went into the system, sampled, and after 3 years of data, conclusively found the exact opposite of what I'd been expecting. If you sample any fish from the main stem of the river that receives effluent from the mine, you find no parasites, none, zero. It was the complete opposite of what I was expecting. But yet, if you travel up into the populations of fish that live up in the Northwest Miramichi or on either the north or the south branch of Big Savogle, beautiful, healthy parasite populations, absolutely beautiful, right? And so this was a completely counterintuitive result. And as it turns out, when we looked into it, and I did subsequent experimental infections and tests in tanks and so on, we found that, as it turns out, we thought that the fish was going to be the most sensitive part of this ecosystem, but as it turns out, the parasite was the most sensitive part of that ecosystem. The parasite was way more sensitive to metal contamination than the host. And in fact, it was so sensitive that we began thinking maybe we can use the parasite as an indicator of metal pollution, not just in this system, but potentially in others, right? So I had to shift my thinking completely from thinking of the parasite as being this opportunistic thing that takes advantage of the poor host that's stressed, and in fact thinking, oh, it's actually the poor parasites that are getting knocked out by this by pollution and so on. And it was a really sort of radical sort of shift for me to have to sort of think about that. You'd think I'd sort of start thinking that way coming out of my master's, but coming out of my master's, I was still sort of thinking of parasites as being just one of the many stressors that affects the host. And I wasn't thinking of the parasite as a creature on its own that is sensitive to environmental disruption and that is sensitive to environmental change. And so really just to make you feel good, just if and when you start your graduate careers or you start doing research and so on, just remember that a guy sitting in this chair is now had this job for now 15 years and so on, had an early career that was absolutely marked by failure. My early career, or where great ideas went to die. My first idea as an undergrad: parasites cause stunting in heavily infected pike in Lac Altet. Wrong. Parasites and pollutants decrease condition and meat quality in Arctic char and igloolik. Wrong. That ended up being, I just didn't, I wasn't able to detect that at all. And then for my PhD, do parasites abundances increase in fish exposed to pollution? Also, no, right? And so I look back on it now and I laugh because it's like just one failure after another. But as you'll see, I learned an important lesson from this, which is that every single one of these failures ended up leading to a more interesting opportunity down the road. And I look back and I think, okay, so if I had gotten a positive result, say from the bottom study, the parasites increase in fish exposed to pollution, if I had gotten a positive result, I might not have been as motivated to continue exploring. I might have gone, okay, well, I got what I expected, so right. But instead, I got something unexpected, and that caused me to think about the system differently. And it motivated me to learn more about the system and think about it differently. And I now think about parasites very differently and much more, I think, intelligently than I did. And I only got to this point because of my failure. I don't think success would have helped me learn as much. So I came out of the PhD, and as a result of networking and talking to everybody and so on, I connected with an absolutely wonderful person with whom I still work, and I still consider him a mentor. Even after all this time, even though he's retired and I've been doing this for a while now, I still think of him as kind of my boss a little bit. And so this is David Marcogliese, and he is the person I did my postdoctoral, my NSERC um, industrial postdoctoral fellowship with at Environment Canada in Montreal. And with him, he was 100. He'd already made the transition that I took forever to make, and he'd already thought about parasites as living organisms that have their own complexity and so on. And under him, I did a bunch of interesting research. For instance, I wanted to see if the pattern that I observed in my master's and my PhD held generally true for most parasite groups in most studies. So I went and collected 300 plus studies from the literature and put them into a giant database and used what is called a meta-analysis, excuse me, a meta-analysis to analyze the data. And the idea here is if you look at this plot, I calculated what is called an effect size. And if the effect is negative, in other words, this is the zero line, you can see the zero line vertically in this plot. And if the effect is below that line, then the contaminant is having a negative effect on the parasite. And if you look, I detected that I figured out through this analysis that the pattern that I'd seen in my master's and my PhD actually holds true for almost every study in parasites. That in almost any study that's bothered to look at it, when you put them all together, show that pollutants have negative effects on parasites. And it doesn't matter whether you're looking at parasite populations, so how many parasites there are in a population, or parasite communities, how many species there are in that community. It doesn't matter the type of parasite, whether it's a simple life cycle or it's a complex life cycle. It doesn't really matter if the parasite is directly or indirectly exposed to the contaminant. So whether it's on the surface of the host or inside the host, and so on. The insight that I'd gotten in my master's and my PhD actually ended up being a generalizable thing that I wrote up in a paper that was published in the journal, a journal called Aquatic Toxicology. And I think to this day, this remains my most cited paper, right? A paper, again, the entire idea for this paper was born out of failure. Initial failure. This paper was a success. You fail enough times, you end up succeeding. So yeah, so I spent 3 years working under Dave Marcogliese, and it was a wonderful experience, and it helped me really sort of shift my thinking about parasites. And Dave had already established himself as a leader in my discipline, and also in generally in ecology by helping try to convince people in other disciplines like in ecology or like in environmental impact assessment and so on, to convince those people to start caring about parasites. Our gut, believe me, I get it. Our gut reaction, if I show you this image here, right? The gut reaction to this image is disgust, right? It's normal. We evolved over billions of years to be averse to certain types of imagery, certain types of visual stimuli, and this is one of those that even parasitologists might find a little bit gross. But what Dave and myself and people who feel the way we do it, what we're trying to get people to understand is that parasites are not only normal, healthy parts of ecosystems, but they are actually critically important for many ecosystems. There are entire ecosystems that fall apart if you remove the parasites from the equation. If you remove the parasites from the equation, maybe an entire group of hosts that are normally controlled by that parasite in the same way a predator would control a host population. If you remove the parasite, the ecosystem falls out of balance and crashes. You have parasites that shift energy flow by castrating certain hosts or killing certain hosts. They shift energy flow in a way that we find beneficial. There's some fantastic work that was done in California, for instance, that found that that parasites shape ecosystems in a way that we find desirable by shifting energy towards fish and towards birds. And otherwise, that ecosystem would have like a lot of things like snails and so on. Instead, you have this much more diverse and much richer ecosystem. And so it's led to these beautiful publications now where our entire shift, there's been this paradigm shift in ecology and in parasitology, shifting away from thinking of parasites as the enemy and thinking of them as natural, sort of healthy components of ecosystems, right? So you see a paper like the one on the left is a healthy ecosystem, one that is rich in parasites. Um, on the right, that's one by my boss, my old boss, Dave. Parasites of the superorganism are parasites indicators of ecosystem health. And you see what a radical change that is from thinking of them merely as disease-causing agents. Now, I will say this. This is not to say that parasites are not causing disease worldwide, and certainly those that are causing disease worldwide should be studied, and we should work towards controlling them or minimizing their effect on humans, and developing treatments and developing preventative measures and vaccines and things like that. As a general rule, from an ecological perspective, parasites are critically important and serve as ecosystem engineers. They're very, very important in ecosystems. So before I launch into the sort of next phase of the talk, I want to recap some of the key lessons that I've learned because the people who are attending this, you're considering what to do next, and you're considering a master's, and that master's is might take you down a research path. And even if it doesn't take you down a research path, you are still going to be doing researchy things and working with mentors and developing ideas and learning new things and so on. And so I think the lessons that I'm saying here, while they are sort of primarily designed for research oriented master's students, I think that they're actually quite universal to anyone considering pursuing graduate work. And so the first one I'd say is that it's okay to not have a plan. I mean, I I turned out okay. I think I mostly turned out okay. I've ended up leading an absolutely incredible life, full of travel and interesting work and brilliant colleagues and interesting discussions and frustration and failure. But remember that that's good, right? So it's okay to not have a plan, and it's okay to just do something because it's interesting or it sounds like it'll be fun. Too often we feel that everything that we do needs to have a purpose or a use, but it's okay to just do something because it's interesting. Um, the second lesson I learned is to not be afraid of failure. Um, Failure isn't fun. Uh, Having a prized hypothesis, a cherished idea get torn up by reality and chucked in the bin is a hard lesson. It's a humbling lesson, but I have learned over time that failure always leads to much more interesting insights than success. Again, if you succeed, then it's just like, okay, well, that worked. Now what? Right? But failure is like, well, why? Why did that fail? Why is my prize idea wrong? Right? Or maybe I tested it wrong, or maybe So this failure is more interesting. The last one is, and this is critically important, no matter what. And it applies to jobs and it applies to research and finding a mentor. It's focus less on what you're doing and more on who you're doing it with. It's way more important to be surrounded by supportive people who are going to elevate you and listen to you and hear you out and support you and criticize your ideas if they don't have a good foundation and support you and motivate you and so on. And so it's very important. Find yourself. It literally, I would say, at the master's level, it literally doesn't matter what your research is, right? I mean, I kind of ended up blundering into a master's project that was parasite related, and I have stuck with the parasites ever since. But most of the people I know don't follow that path. They do something in their master's, they end up doing something different in their PhD, or their undergraduate project was in one thing, and they do end up doing something else. I mean, I think about my own dad, whose undergrad experience was, I guess, geology and then economics, and then he did an MBA, but his first job was with IBM, right? So it's like these people's path is never linear, right? So it's much more important to just keep moving forward and make sure that you're doing it in the company of people who are supporting you and understand you and will support you in your aspirations and your dreams. Which is why I feel so lucky because I ended up blundering into a bunch of really cool partnerships here at Nova Southeastern. And so I've been in Nova Southeastern for 11 years. No, sorry, since 2011. So about 13, 14, 15 years or so. And in that time, I have been so fortunate in the number of people who have looped me into their work, and it's been such a privilege to be part of their thought process and their labs and to work collaboratively with them and so on. And I have so many that I could spend the next hour listing them. But I'm going to focus on the three that have probably ended up we've ended up working on the most students together. And so the first one I want to mention is Robert Smith, who's over at KPCOM with the medical college. And he and I have done such interesting work on parasite bacteria interactions and nematode behavior and how you can engineer bacteria to potentially control the soil stage of parasitic nematodes. So absolutely really cool research project. As you can see, he's a fellow Canadian. Excuse me. But that's Rob Smith. I continue to interact with him, and hopefully we will continue to work on projects in the future. In the middle, you see David Kerstetter, who I think I would describe as my main research partner here at Nova Southeastern, and he another interesting background is background in political science, but ended up moving towards fisheries management and fisheries, and is now interested in ecology and birds and coastal wildlife and things like that. So another really interesting, really twisty and turny um, career path. And so my research aligns really well with Dave. Dave studies these organisms and gets access to incredible samples, and I help study the parasites that are in those samples. And so together we've ended up supervising a number of students who've done really cool work on the parasites of Florida wildlife, including birds, fishes, and many invasive species. Including things like lionfish and iguanas, which, if there's time, I'll talk about at the end of this talk. We've also had the chance to work on parasite ecology in critical Florida habitats like mangroves and seagrass and coral reef systems and so on. So that has been another really fortunate collaboration. And then the third person that I'd mention, and again, this is just a sample of three people that I think I've worked with the most, is Sean Locke, a great friend of mine. We did our postdoc together with Dave Marcogliese at Environment Canada, and we've continued our friendship and our collaboration since then. He's now at the University of Puerto Rico, and he studies parasite phylogeny and the parasites of Caribbean wildlife. And we have found many opportunities to work together and co-supervise each other's students and so on. And it's been an absolute wonderful thing. And he's introduced me to the wonderful world of molecular biology and molecular phylogeny and DNA extraction and comparing sequences collected from different organisms to determine their evolutionary relationships and so on. So yeah, I've been lucky, and again, I could list another dozen people who I continue to work with, and I'm so fortunate. And remember the lesson that I gave you at the end here. It's really who you're doing it with that's important, and in that regard, I could not be luckier. I've led a very fortunate life in the the types of people I've had the opportunity to collaborate with. I've also, and this is going to be of direct interest to you, had the opportunity to supervise some absolutely fantastic students. And again, there's I think I'm up to 35 or 40 now that have moved on. And it's an absolutely bonkers number. And I'm just going to pull out a few that are just as a sample of some of the things that our students have that come through the lab and who are doing interesting things. So for instance, I'll start with Kaylin Simmons, who was mine and Dave's first master's student. And she did her master's at Nova Southeastern. She did a PhD, I think, in Atlanta, and is now an oceanographer with NOAA. So living the dream, absolutely fantastic career. In the middle, you have Dana Hun, and Dana Hun is did her master's with us, and she is now with the Florida Fish and Wildlife Conservation Commission. And the story with Dana, is one of the things I really love about where I am as a scientist is that some of the students that we've trained are now looping back and are now in positions where they're doing interesting work and are now becoming research partners, right? And so they're no longer like students, they're peers, they're collaborators, they're colleagues, and we're working together and trading ideas about cool projects and so on. And so that's exactly what's happening with Dana. We've got multiple projects on the go potentially with Dana. starting up. So it's just one of the wonderful things about doing this job is watching people grow and find and follow their dreams and then eventually become potential research collaborators. And then on the right, Matt Woodstock, who did his master's with us, did a PhD down in Miami, and is now a postdoctoral research fellow at the Woods Hole Oceanographic Institute. So you can see some of the wonderful places where our students end up. Just a couple more, just because I know some of you are more interested in the in the medical side of things. A couple of examples of students who've taken their study in my lab and then moved on towards more medical pursuits. On the left there, you have Hema Narlapati, who did some work with us on the work uh, with Rob, looking at engineered bacteria and nematode behavior and so on. And then that's her with the big galoot in the middle is this guy called John Hodden, who's a parasitologist at George Washington University, and this is a very hairy version of me here on in the turquoise shirt. And Hema is now in Hawaii, and she is at the Tripler Army Medical Center. She got her DO. So she's a doctor of osteopathic medicine, and she is a specialist in internal medicine at the Tripler. And so she's doing incredibly well and doing wonderful things. She's um, um, and then on the right is Jacqueline Santis, who worked in our lab on and the iguana project. I'll talk very briefly about because I do have a tiny bit of time. Um, and so Jacqueline is one of several students who worked on that iguana project, and we're still in touch because we're writing that project up to be published. And but she graduated as a registered nurse, and she is now a specialist at the cardiothoracic unit at the University of Miami Hospital. Right. So this is just two that I've pulled out that quickly sort of that came to mind. I could list another two or three, but don't want to don't want belabor this too much, but just to give you an idea, like the students that come through here, um, always it's find value in it, and I'm always amazed at some of the interesting things that they go on and do. It's there's almost this weird sort of I don't know how this is going to sound, but it's a weird sort of proud parent sort of feeling to watch these young people come into your lab and they don't know how to do anything and they break everything. And then by the time they're done, they're so good at what they do and they're so amazing and they go on to do so amazing things and they're so confident. I love it. It's fantastic. So yeah. And so what are some of these opportunities and how have these opportunities ended up sort of continuing to yield interesting insights? I was sort of struggling to figure out which project to talk to you about. And what I've decided to do is pull an ongoing project that just highlights how surprise and the unexpected can yield really interesting insights. Um, so I'm going to talk to you a little bit about one of the several ongoing collaborations that are going on in my lab, specifically the study of parasites of iguanas, which are an invasive species here in South Florida. And this is work that I'm carrying on with a bunch of people, such as like Dave Kerstetter, who I mentioned earlier, and Andrew Osga, who's a biology professor who also supervises graduate students and so on. Um, and so the way that this project goes is that there are these iguanas in South Florida. We were interested in looking at their parasites and seeing what parasites they have that might control them, and do they have any parasites that they might be introducing into Florida that might affect native reptiles. So that was the main reason why we started doing this. And we dissected a bunch of iguanas and found that they have almost no parasites. The only parasite that we find consistently is a worm that's in the digestive system of the iguana. So here you can see on the right this picture of the an excised part of the digestive system. Here you can see a partially digested. This is probably almost about to become poo here, and you can see that this structure called the cecum, which is like a bag in the intestine, where a lot of the digestion happens, is packed to the rafters with these worms. So all this sort of spaghetti stuff, those are the worms. All right. And this is the only parasite that we ever found in any sort of meaningful quantity in these iguanas. It really seems to be just this and nothing else. So I decided let's have a look at these worms a little bit more closely. We identified the worms as something called Ozolimus megatyphlon. They're actually quite large. And the literature on this parasite is unequivocal. It is consistently described in the literature as a parasite that causes harm to the iguana. And in fact, the insert that I put here from this journal, the Japanese Veterinary Medical Association here from 1999, shows how many worms were recovered from an iguana, and the authors conclude that the iguana was sick and that the iguana was sick because of these worms. But the problem is that every iguana that we've found, healthy, beautiful iguanas, females with tons of eggs, males very clearly able to defend territories, are packed to the rafters with this worm as well. So I didn't really believe that this worm was as pathogenic as the literature seemed to suggest. So while I was pondering that, one of the students that we had working in the lab said, Dr. Blanar, what are the hairs on the surface of this nematode? And I was not looking and I was very dismissive. I was like, nematodes don't have hairs. Like, don't be ridiculous. Nematodes don't have hairs. And he was like, no, dude, seriously, this nematode is hairy. Come have a look. And he pointed out there are actual little hairs on these nematodes. And I was like, what? What? And we tried to scrape them off. I was like, maybe it's fungal growth, maybe it's something else. And they were actually embedded in the cuticle, the skin of these worms. So we decided to do some scanning electron micrography. And as it turns out, the little hairs that are on the surface of the worm are actually these filamentous bacteria that are embedded in the cuticle. And so that's what you see in this microscope image here. And then if you zoom in a little bit further, you see that living among this forest of filamentous bacteria, you have this incredibly rich, incredibly diverse community of bacteria. And in the same community, you don't see the same community on the walls or on the surface of the cecum of the actual host intestine. Most of the micro community in this system seems to be bound to the surface of the worm. So I was like, what is going on here? Like, this is such a weird result, and so on. So we did some DNA extractions and we sent the samples in for analysis. And I guess I'll just give you the quick version of this. On the left, you have the microbiome. How to read this plot. On the sort of vertically, you've got all the different kinds of bacteria that you might find listed here. And then the boxes, if the box is really red, there's a lot of that bacterium. If the box is really blue, there isn't a lot of that bacterium. And the one thing I'll point out is that the nematodes have the worms have a very distinct microbiome on their surface than the iguana gut does. And if you go in and you look at what these bacteria typically do, they're fermenters of plant material. So this thing, this worm that is inside the gut of this plant-eating reptile that fills this digestive cavity like this is covered in microbes that help digest the plants that the iguana is eating. And so now we're coming around to not thinking about this thing as a parasite at all and disagreeing with this literature completely, but actually wondering whether these nematodes, because of the microbiome that they have on their cuticle, might actually be beneficial to the iguanas. And that completely throws upside down the whole idea that these things are parasites, because a central part of how we define a parasite is that it causes damage to the host. It exploits it, it kills it, it harms it in some way, it negatively affects it, it causes disease. And yet here we have a parasite, quote unquote, that actually, by all measures, is probably benefiting its host rather than harming it, right? So this is just an example of like the crazy sort of bonkers insights that you can sometimes get, and the sort of unusual pathways that my students' research has taken me. This whole iguana project was born because a single student was looking at the worms and said, "Hey, Dr. Blanar, they're hairy." And I said, "No, they aren't." And he said, "Yes, they are." And I had to again go through that process of being wrong, and that led to the more interesting insight. Okay. So it's 44, 45-ish minutes in. I think I've probably said enough. I'm hoping that some of you have questions. I'm here to answer them. And of course, I'm sure you have many questions about the masters in general, and I'll be happy to sort of turn over the microphone to my colleagues from recruitment. But if you have any questions about the talk itself or any of the parasitological work that I've been doing or any opportunities, I'd be delighted to talk about them. So thank you very much for your attention, and I'll turn things back over to Farah and Megan. Thank you very much.
Dr. Blanar, that was very interesting. I wish my children were watching this. Um, I hope our attendees actually enjoyed it as much as I did, and you offered great advice to everyone. So thank you for that. We will now take any questions you all might have. If you still haven't done so, please write your questions for us in the chat box and we'll answer them as they come in. We do have one that came in while you were talking, Dr. Bonar, and it says, are these parasites zoonotic?
So yeah, that's such a great question. So just for the audience who might not be familiar with this terminology, a zoonotic parasite is one that goes from wildlife into humans potentially. And so the and this doesn't just apply to parasites. There are bacteria and viruses that can also potentially be zoonotic as well. In fact, we closed down for several years in 2020 because of a zoonotic virus that jumped into humans from wildlife. So yeah, some of the parasites that I've talked about are zoonotic. The tapeworm that I talked about that I studied in my master's is definitely zoonotic. There are definitely instances of humans getting it. The one that I studied for my doctoral, no, that one is strictly a fish one because it binds to fish gills. And in fact, only binds to, as far as I know, to salmonid fish gills, so fish in the salmon family. Um, and but yeah, there are a great many zoonotic parasites, including some of the ones that we study in our lab, right? So there, I think that the we really haven't delved too deeply into an analysis of which of the parasites we're finding in our local birds or in our local fish have zoonotic potential, and that would be a really interesting research study. In fact, it's such a good idea, I might just write it down. But it's really, it'd be really interesting to do that kind of a survey.
Thank you, Dr. Bonar. Another question, how was your experience at NSU different than that at all the other universities you worked at?
Well, for one thing, I was a boss. I got to make the decisions rather than doing what other people told me to do, which is a great and liberating experience, but it's also terrifying because you don't have anyone to blame if things go badly or if things stall out. The buck definitely stops here. And any, it's like when people say, oh, any mistakes that are made are not my students, but my own. My experience at Nova Southeastern it has been really good. I've had the chance to work with some really motivated, really interesting students, but that's been true everywhere. I'd say the biggest difference for me is just how much I've had to learn and continue to have had to learn since I've gotten here. You have to remember, I'm Canadian. I'm used to working in Canadian systems, which are a bit more, and in rivers and so on, which are a bit more tractable. And here, suddenly, I'm working in Florida with so much more biodiversity and in marine and coastal systems that are inherently complex and so many different additional. I used to be just a fish guy, and now I'm a bird guy, and I do reptile stuff, and so it's a lot more complicated. And it's wonderful because it's never been boring. No 2 days are the same. I have had to learn every day of my life since coming here. I've been on the steep part of the learning curve, and that's actually really cool because there's absolutely zero chance I'm going to get bored, and I see no end in sight in terms of when these learning curves are going to end. So that's been, I think, the biggest change is coming down here, and the systems are just so much more complex.
Dr. Blanar, what qualities are you looking for in prospective students who want to work in your labs?
Curiosity, um, independence, but curiosity and a willingness to try new things, interest and an interest in what they're actually doing. I think the most important thing whenever I talk to a student is the student has to be sort of legitimately, I don't know how to say this. It's like what I said about the the it's not so important what you're doing is who you're doing it with. And I think that the student has to be sort of legitimately interested in learning. I don't expect the student to know very much about parasites and parasitism because let's face it, there isn't a lot of parasitological training out there. And very often students come into my lab and need to learn that stuff from scratch, and that's fine. That's kind of what I've gotten used to. Every now and then I get a student that comes from a parasitological lab, and that's great. They can hit the ground running. But really, it doesn't matter how much prior experience you have. You have to come in with a good attitude, and you have to be willing to ask questions, and you need to be self-motivated and so on. So yeah, those are some of the qualities I like to see in students.
So the student that asked you about the zoonotic parasites, he actually lost connection and was not able to hear the answer. So if you can briefly explain to him again.
Yeah, sure, George. Yeah, so the answer is that the iguana parasite is not going to be zoonotic. The only parasite that I've studied that really has any sort of zoonotic potential was the tapeworm that I studied for my master's. Um, and so that one there definitely gets into humans and causes disease in humans. That said, part of my answer was to say that we've really not done a huge inventory of the wildlife parasites that have zoonotic potential in South Florida. And I was saying that that would actually make a really good research project, and I'm totally stealing the idea. So I've already written it down. It's a really cool idea to look through some of the things we've already found and think about which ones have zoonotic potential.
Another question, is it okay not to have a specific research interest or focus when applying for a graduate school? If it is okay, does it not reduce your chances of being selected onto a team?
I'll go ahead
I
and take that one.
Okay, go ahead, Megan, for sure.
So I work directly with Dr. Johnston, who is over the students that are in our research portion of our master's degree program. And for the students that aren't really sold on their specific research interests, there's still opportunities for you to join. One of the things I recommend, though, is that you do look into what our faculty is currently researching and see if any of it piques your interest. We ask when you're applying that you tell us a little bit about the things you are interested in. So it's not that you have to be set in stone. You don't have to have an exact topic, but it is good to kind of know where your interest might lie. You have about 2 semesters into the research focus to make a decision on what your thesis or capstone project will be on. So again, you don't have to come in knowing exactly what you would like to research, but it is good to narrow down the areas of focus that you would like to possibly pursue. That way you can consider reaching out to those faculty members to maybe work with them on some of the research they're currently doing. Maybe it might get you an opportunity to work in a specific laboratory, but no, you don't have to have a specific topic in order to get into our master's program.
I think that's such that's so true. And the one thing I'll say, and this is the props to the recruiting people, is that recruiting people do a number of really good events that allow students to potentially meet and interact with people who might be interested in supervising students. And you should attend every one of those events. It's a really good way to meet potential faculty and interact with them. And remember that for me, the game changer was interacting with faculty in an unusual sort of outside of the lab sort of way. And that was the moment for me. So definitely any events that these good folks organize, please make sure you attend.
The same applicant or the same student actually who asked you about the zoonotic, he's saying that he'd be honored if you consider him for this potential research topic. So you've already got
There you
someone.
go. See? Yeah,
Um,
that like I said, it would make a really cool, it's a cool idea.
we have a question. Have you looked into, and I hope I say it correct,
Elasmo Brank.
elasmo branch parasitological systems before? And if not, is it something you would be interested in?
Yeah, we have. And in fact, So we have, for instance, a current student, Sarah Sargent, is looking at parasites of stingrays in the Indian River Lagoon. We've also had students do, I think that might be it. Yeah, I think the Stingray project is our big sort of introduction to the thing. If David, I don't know if Dave's is in the crowd. If I'm wrong, and then pop into the chat and tell me that I'm a fool, and I'm forgetting something. But one of the things that's really neat about the elasmobranch parasites is that they have larval stages that are very commonly found in the bony fish that we study. So even though we're studying bony fish, a large proportion of the parasites that we're finding, especially the juvenile parasites that we're finding in the bony fishes in these reef and coastal systems, are ultimately going to reach adulthood in sharks. So even indirectly, we kind of end up studying shark parasites. It's actually really cool. Yeah, so the answer is yes, but we're really only just starting that aspect, I think.
Thank you. I'm looking to go into veterinary parasitology research. Do you have any advice on how to get into parasitology research after undergrad and before vet school?
I mean, the masters would provide an opportunity to do that kind of thing. And the cool thing about it is that the way that we're set up in South Florida is that if you're interested in veterinary parasites of horses, there's horses all around the main campus, and I have connections to people who work with horses and regularly have to deworm them, and so there might be a potential study on sort of some of the transmission dynamics of these parasites here in South Florida, which I don't think are very well studied. There's also canine hookworm, which I think is grossly understudied here in South Florida. I have a sort of a longstanding sort of curiosity about other sort of things like Angiostrongylus, which is a rat lungworm, getting into cats and dogs, but especially cats, and so on. So there are lots of potential possibilities of research that are directly vet related down here. So yeah, I think it would be a good potential thing. You just need to sort of I don't know, discuss possible ideas. We need to see what is possible.
Okay, thank you, Dr. Blanar. That was our last question. There's a question from us, the graduate admissions team. What would you tell our prospective applicants who want to apply to NSU but are still unsure if this is the right program or the right university for them?
I would. Sorry, go ahead, Megan.
I would recommend, just like Dr. Blanar said earlier, is to continue attending some of the events that we host. For the Department of Biological Sciences in particular, we'll be hosting a virtual open house coming up in about 3 weeks. You should all receive an invitation to that as attendees of this event. What's great about a virtual open house is it gives you an opportunity to hear from more faculty about some of our programs, but also it gives you an opportunity to hear from current students and alumni of our programs. So you get to listen in from a student perspective. Some of our most recent alumni are going to be presenting their experiences in this upcoming open house event. But not just that, I usually tell students, look at a couple of programs, see what they all offer you. Big plug right here. One of the things that we do offer is our students an opportunity to be teaching assistants. So not only can you come to NSU and get your master's degree in a subject that you're interested in, but you also have this opportunity to become a TA. That gives you a little bit of a boost on your resume if you're looking to apply to postgraduate doctoral programs, if you're looking to possibly go into academia. That gives you an opportunity that you might not have at other institutions. So that is a great thing. But doing research, attending events just like this is always going to be helpful. No one can determine but yourself whether or not an institution is a good fit for you. I do think our program is great, and our alumni go off and do some wonderful things. So I encourage you, when you get the invitation to open house, to go ahead and register for that event as well. That way you can get a little bit more insight from other faculty members and alumni and current students.
Thank you so much, Megan. Thank you so much, Dr. Blanar, for sharing this experience with us all. We truly appreciate it. And again, Megan, thank you. And, you know, all the graduate admission team that facilitates these events for our students. If you're one of our students, yay to you. If you're still not one of our students and would like to be, every one of us here at the Graduate Admissions Department is eager to help you be an NSU Shark. On the screen right now, you can see our contact information and social media account that you can follow us on. And this is where we advertise for our upcoming events and open houses and lectures like these. With this, I end our lecture for today. Have a good night, everyone. Thank you.